Good afternoon, and welcome back to our monthly Living with ALS webinar series. I am Lori McFerrin with the ALS Association, and I am happy to be hosting today's conversation with Dr. Kuldip Dave. Before we start, I want to share that this and each monthly webinar is being recorded and will be posted on our website within a couple of days. We will have time for questions after our presentation. Add your questions in the Q&A or the chat feature. Hopefully we'll be able to get to everyone's questions in our time together today. I'll also ask you to stay for just a few minutes following today's presentation to complete a brief 3-question survey. Your feedback lets us know if we're meeting our educational objectives and also helps us plan future webinar topics. So thank you in advance for your help. Based on some of your recent requests for research updates, our time today will focus on research, specifically antisense therapy. Last year, the Food and Drug Administration approved the first ever genetically targeted therapy for ALS, which served as a promising endorsement of the antisense technology that underlies its use. So how do these technologies work? And how are researchers using it to develop new treatments for people with genetic and non-genetic forms of ALS? Luckily, we have Dr. Kuldip Dave, Senior Vice President of Research at the ALS Association, with us to answer these questions. In this role, Dr. Dave directs the association's research strategy through research funding, research programs, and other strategic initiatives. Sharing a bit about his background, Dr. Dave received his bachelor's degree in biology from Rutgers University and a PhD in pharmacology and physiology from Drexel University College of Medicine. He completed a postdoctoral fellowship at a biotechnology firm and later joined the pharmaceutical industry. He went on to work for the Michael J. Fox Foundation for Parkinson's research, and after 9 years there, joined us at the ALS Association. He's been with us since May of 2019. Dr. Dave, welcome. We're happy to have you here with us today.
And thank you for having me. Hi, everybody. Today I'm going to talk about a very complex topic. And so I really appreciate the title of Making Sense of Antisense Therapy. And I'll talk about why this type of therapy is really groundbreaking right now and what it has meant for the ALS community. But before I do that, I think it's important to talk about some really basic genetics and understanding of genes and how did we get here. And so to do that, I will talk about sort of a very high level what we think is sort of genetic ALS, or the component of genetic or familial ALS. So let's just kind of take a broad view at first. ALS is, we think there's more than 30,000 people living with ALS right now. And if you look at the breakdown of people that have ALS, because we don't know why they have ALS, what we call sporadic or idiopathic ALS, in that the cause is unknown. And that's about, makes up, as you can see in this chart, 90% of the pie, we think. By the way, these numbers are high-level rough numbers. You could argue it's 88% or 92%. So, to make it easy, and at least that's where the academic community is right now, let's say it's 90-10. And so 90% of it is where we don't know what the cause is. It could be because of, toxin exposure. It could be head trauma. We know military service increases your risk of getting ALS by 2-fold. This is what the CDC registry is doing. They're collecting information on where people live, what people do or people did, and trying to make sense of what could be that question mark on idiopathic, what could be the cause, and it could be different for different people. However, while that 90% is unknown, 10%, we have a pretty good idea what that is. And that is what we call familial ALS. And that is that a person has a specific gene and that there are family members who have been diagnosed and probably that gene has been inherited. And that's why we call it familial ALS. And if you look within, so here's a pie, you see 90%, you see 10%. Now, if you blow up that 10%, and you say, what constitutes that 10%? What type of genes there are? And you could see the breakdown there. There is about 40% of that 10% is mutations in genes for C9orf72. C9orf72 is a protein that's involved in trafficking and handling of other proteins. And by the way, in the next slide, I'll talk about sort of what proteins are and how, what that means. But just giving you a sort of a high level sense of that. About 20% of the familial ALS is SOD1 or SOD1, and that's an antioxidant protein in our cells. And it essentially protects us from toxins inside the cell. This is also a really important protein because tofersen, that was approved by the FDA last year, the first ever antisense therapy in ALS, targets this protein, SOD1. And then you can see there's 5% for FUS protein or FUS protein. And that's a protein that's involved in DNA and RNA repair. And so point of this is that while we don't know about the sporadic piece, the familial piece, we have a good sense because we know the genes that have been linked. And before I talk about what genes have been linked to ALS, I think it's important to talk about a very simple fact of what DNA, RNA, and protein is, because we're going to make sense of antisense therapy. If we are to understand antisense therapy, we have to understand what these three components are. So what normally happens is that, and I hope you all can see my pointer here, but normally when you have no mutations, all of our cells, every cell in our body has DNA. And this DNA is really our genetic code. It is a code to make every single protein in your body. Every single protein, whether it's skin protein or hair protein or nail protein or proteins that make up the brain cells or any cells in your body. Every protein in your body has a code written in your DNA. And so think of DNA as ingredients to make cake. I'm going to make you all very hungry after lunch now. So, what do you need to make cake? Flour, butter, eggs, sugar. Let's make it simple. Those four things. Those are the ingredients. That's what the DNA is. The DNA is sort of your ingredients of how to make proteins. Then you get what's called a messenger RNA. So from a DNA gets transcribed into messenger RNA. Messenger RNA is essentially the instructions or the recipe. So this RNA now takes the DNA and makes a protein out of it. So it makes the cake. So DNA is the ingredients, RNA is the instructions or the recipe. So RNA basically takes all that information and makes a protein, or in this analogy, a cake. And so every protein in your body is made in this way. It goes through a DNA to RNA or messenger RNA, and then messenger RNA to a normal protein. And you could see the protein, it folds in a certain way. It kind of takes a its own structure. And if everything goes right, if the DNA is all good, if the mRNA is all good, you're going to get the right protein almost every single time. However, sometimes you have a mutation in your protein, in your DNA, excuse me. And you could see that on the right here, the mutated DNA. And this is where, and it's shown here in red. And so this could be mutation because it's either passed down from your mother or father, or it happened randomly during development. That happens. Those are sporadic mutations that sometimes happen even if there is no family member with that mutation. And so that happens. And so when these mutations happen, what happens is you get a mutated mRNA. And the analogy I would like to use is, think of if instead of sugar, there was salt present. So now you had flour and butter and eggs, but you had salt instead of sugar. And so now the mutated RNA is taking that ingredient and developing a protein or a product which is not really cake, or it's not going to taste like cake. It's not going to be the right protein. And sometimes that mutation may cause a protein that doesn't affect you, but sometimes it could cause a protein that is toxic. And that is essentially what happens in ALS. We think that mutations in genes produce toxic proteins, and these toxic proteins then lead to, amongst other things, death of neurons or motor neurons, which is the type of neurons that die off in ALS. And so this is really, really important to understand. If you take anything back today, it's this, that DNA goes to RNA, RNA goes to protein. But if there's a mutation, your ingredients are not right. And so the recipe is going to take those ingredients and develop something that you're not going to like. It's going to be messy. It's going to be non-functioning, or perhaps worse, toxic to the cell. So now that we have understood the basic genetics piece, I think we can start getting into, remember I told you there are some genes that are associated with ALS. Well, you may be wondering how many genes are associated with ALS. And there are about 40 different genes associated or linked with ALS. And this is a really, really difficult figure to assess. So I'll give you some take-home messages here. First, you could see down here, year discovered. So when was the first year when we discovered a gene that was linked to ALS? Was 1994, when we linked SOD1 or SOD1. It was the first protein linked to ALS. And the other take-home message here is, look at between 1994 and 2012, I would say 2014. You see sort of this increase, but the curve was slow discovery. But as 2012, 2014 happened, you could see that the rate of discovery has just increased exponentially. And there's two reasons for this, really. One is that the technology of genetic sequencing is getting better and faster and cheaper. And so you are able to sequence a whole genome in a matter of days. And you could quickly find these mutations. And so that's great. The second reason why you see this amazing exponential increase is I expected funding. I think our field was lucky because in 2012, 2014 and on, those two things started happening. The technology got better and cheaper, and we got an influx of dollars into our ecosystem. And you could see the amount of proteins that have been discovered in just the last 10 years. A couple of things to note. Another note here is, as you can see there, I'm not going to require you to know all these 40 proteins, but just pointing some of them out to you. there is something called TBK1. That's a protein that's involved in immune system. It's also is involved in kind of handling and processing other proteins. There is KIF5A up here that was discovered in 2018. And that's a protein that's involved in sort of microtubule and neuronal structure piece. So the point is that we're finding genes faster, more efficiently, and that there are all kinds of different types of genes that are associated with ALS. Okay, so now that you have a basic understanding of, what part of ALS is familial and how many genes constitute that familial ALS, 40, and now that you have learned the cake analogy of DNA going to RNA and RNA going to protein, let's get into the topic of today, which is what is antisense therapy or antisense oligonucleotides. And so an antisense therapy is a way to treat ALS and other neurological conditions using short DNA-like molecules called antisense oligonucleotides, or ASOs. And what ASOs really do is that so here's your mRNA. Remember the messenger RNA, the recipe that I talked about? And assuming that there are some messages in there that are mutated because you had a mutation in your DNA, the antisense oligonucleotide, or ASO, would come and bind that area where there is a mutation. Or it could bind another area where in the RNA and do one of two things. It can either knock down the RNA, knock down as in make it non-functional. So that RNA never makes protein, never goes on to make protein. It's never translated. It can either do that, or you have some really, really amazing ASOs that can actually correct the mutation in such a way that you're forming normal protein. And that's what we call modulated splicing or splice modulation. So really, those are the two ways the antisense oligonucleotides work. And I'll show you sort of a more specific picture on that. But it's the take-home message from this is that it is a drug treatment. It targets messenger RNA. And what it does, it alters the expression of this messenger RNA or change what the messenger RNA is expressing. So either it stops the expression of the bad protein, or it changes the expression of the mRNA in such a way that normal protein is formed. All right, so let me give you some examples of this. And so if you go to the next slide here, this slide does a really good job showing you how a knockdown antisense oligonucleotide works. And so on the left here again, and I'll reiterate these things so that you can remember how all of this happens, right? So you got ingredients, you got instructions, and then you got cake. And so you have your DNA. If it's normal, if it has no mutations, it gets transcribed into a messenger RNA. Messenger RNA gets translated into a normal protein, and your proteins are functioning fine. If you have one of those 40 genes that I talked about in the earlier slide, then you're going to get you'll have a mutated DNA or a mutation in your DNA, which means you now have a mutated messenger RNA. And because of that, you're going to develop a protein, most likely a toxic protein that you don't want. And so what an antisense oligonucleotide, or ASO, does is that it comes in, and very important to remember, it doesn't bind the DNA. it binds the messenger RNA, in this case, the mutated messenger RNA. And in this example, it actually stops that messenger RNA in such a way that it does not make the protein. So in this case, the bad protein is not made at all. And so this is how knockdown antisense oligonucleotides work. By the way, tofersen or Qalsody, the Antisense therapy that FDA approved last year, is a knockdown antisense oligonucleotide. It knocks down SOD1. And I have a slide on that to show you later. So while this is one way to use an antisense oligonucleotide, there is another way to also go after mutated mutations, and that's called the splice modulating approach. And I'm not going to really walk you through this. I think just kind of important to, I'll just kind of do a very high level on this, is that what normally happens is that you have, from the DNA you get, and I didn't really show you this, from the DNA you get this pre-mRNA, and then you get mRNA, and then you get protein. And what happens in the between pre-mRNA and RNA is that you get these areas called introns taken out, and then these exon areas are pulled together, and then these exon areas of the gene then form the normal protein. Sometimes what you get is abnormal splicing. So you get places where splicing happens in such a way that you could see, instead of it getting 3 exons, you get 2 exons, an intron and an exon. So you could have, this could be another reason why you could get abnormal protein. And so in this case, what an antisense oligonucleotide would do is bind to the messenger RNA, or the pre-messenger RNA in this case, and make sure that the right exons are brought together, and so that you get normal protein. And so there are kind of 2 ways that this could work, as I said again, a knockdown approach where bad protein is not made, or a modulating approach where you modify the RNA in such a way that normal protein is now produced. And so these are really the 2 ways to know about how RNAs work. Now, you may be wondering if are antisense therapies only being developed for ALS? In fact, they're not. There are several antisense therapies that have been approved in the United States, both for ALS, as in tofersen, Qalsody, but also there are other diseases such as Barton's disease or spinal muscular atrophy, where you have nusinersen, which was approved by the FDA a few years back. And so this is not just an emerging area in ALS, but neurological diseases in general. Okay, so let's go to the next slide. So I already set this up for you, and we are very lucky that in the ALS space that we have had a approval of antisense therapy. This happened last year for SOD1 mutations. And this is, if I want to take you back to that slide where you had the big pie with 90% and 10%, it's 20%, remember, was familial. And out of that 20% was SOD1. And so numbers are low in terms of how many people this therapy will treat. We think there's about 400 to 500 people in the United States that have SOD1 related ALS. However, this approval is really important. So tofersen or Qalsody, developed by Biogen and Ionis, received an accelerated approval. Accelerated approval is based on some biomarker that changes. In this case, there is a reduction of neurofilament light, and this neurofilament light reduction was seen as a biomarker that was able to reasonably predict clinical benefit. And it was on that basis that FDA approved tofersen in April last year. It targets the RNA, and it is a knockdown antisense oligonucleotide approach, and it stops toxic SOD1 protein from being made. And in fact, we saw that in the phase 3 trial results that were shared, tofersen reduced SOD1 protein levels in CSF or the spinal fluid by 35%, even just 8 weeks after treatment. And so this is really important. The 2 points to take away here that are crucial. One, that the ALS Association was one of the first organizations who funded this at a very, very early stage. When antisense oligonucleotides were being developed in early 2000s, it was not seen as a very tractable approach. But the association was the first one to fund an antisense oligonucleotide development for ALS back in 2004. And that was not the only funding. We've given almost 5 or 6 different follow-on fundings to keep moving that technology forward, to the point that Biogen and Ionis were able to then bring tofersen to the market. And so really important to note that sometimes these approaches are new and novel, and if an association or a foundation is risk averse, you're going to overlook that new therapy or new approach. And we were happy to de-risk this approach and its development. And then the other point to take home is this ongoing phase 3 ATLAS study. And this is really, really important because this is the first ever prevention study in ALS. And what do I mean by prevention? Well, prevention is how do we get people to not get ALS? How do we prevent either ALS from happening in the first place, or maybe perhaps delay ALS significantly? And we're doing this, Dr. Michael Benatar, who also happens to be the chair of our association's research committee, is the PI on the study that he is leading with Biogen. Is testing whether tofersen can prevent or delay ALS symptoms in people that have SOD1 mutations but don't have the disease yet. Again, really important. ATLAS study is testing tofersen or Qalsody in people that have SOD1 mutations but don't have the disease, and that's what we call, or who we call, asymptomatic carriers of the disease. And gosh, if we are able to show that because of the way tofersen works, which is by knocking down toxic protein development, that people would never develop the disease, or we can push the disease back by several years or decade, wouldn't that be just, phenomenal? And that's what this ATLAS study is doing, and this is why genetic testing is so important. And I'll talk about that a little bit later. The other, so while this is an approved drug, and also there's an ATLAS study ongoing, it's also important to think of a current drug that's going on in phase 3, and that is called JC-Fusin. And this is a drug that goes after the FUS gene, or FUS gene. FUS mediated ALS is very rare. It's generally associated with juvenile ALS. You see people as young as in their teens or 20s get ALS. And it's very rapid and progressive form of ALS. JC Fusen gets its name from a person living with ALS, JC Homstead. She was the first person to receive this drug. And it's about, it does the same thing that tofersen does with SOD1. It binds to the messenger RNA, and it reduces production of the toxic FUS protein or FUS protein. And that's how it works. And there is a point here to make on trial is that we, the ALS Association, were the first ones, again, who in this case partnered with Project ALS to fund a feasibility study in a very small group of people living with ALS with this mutation. And only when that feasibility study was shown to work and be safe, that a larger trial that Ionis is now currently doing got started, which is the larger phase 3 ongoing trial. And so this has been a really interesting project to, again, show de-risking. Our funding de-risked, our funding for the early clinical feasibility study de-risked the larger phase 3 trial. And so I will, and that's it on JC Fusen. And so with that, let's talk about the importance of genetic testing. And so why is genetic testing important? Well, one reason why it's important is because how else are you going to know if you have one of the 40 genes that have been linked to ALS? This is why genetic testing is important. What genetic testing does is that it looks for harmful changes in DNA. It looks for those mutations. It's a very simple test. It's done through either a cheek swab or a saliva sample or a blood sample. And it's sent out. And then you generally get a report back that will say either you don't have any known ALS mutations, or that you have one of the 40 ALS linked mutations in one of the genes that have been linked to ALS. And so if you know, what does that mean? If you know, let's say you get a test done, and you know that you have a particular gene. Now, if you have ALS already, then it makes you eligible for an FDA-approved therapy. So, let's say you have ALS and you get tested and you find out that your ALS is not a sporadic, the 90%, but maybe it's that 10%, and maybe you have SOD1 linked ALS. Well, if you have SOD1 linked ALS, then you're eligible to get tofersen or Qalsody that's approved by the FDA. Similarly, if you have another gene, let's say you have a C9orf72 gene, or you have the FUS gene, well, you're then eligible for a clinical trial that's ongoing. There are trials for C9orf72. There are trials that are ongoing with FUS, as I said, for the Ionis large phase 3 trial. These are trials that you can participate in because you have a mutation, even if the FDA hasn't approved those therapies yet. What if you're a family member of someone that has ALS, and maybe you know that they have a particular gene? Well, you may want to know that whether you have that gene. And in the case of SOD1, that's even more important because there's already an ongoing study that I talked about, the ATLAS prevention trial, which is looking at asymptomatic carriers. So if you are a family member of someone that has SOD1 linked ALS, then you may want to get genetically tested because you may want to participate in this trial which may show that you are able to prevent, that we may be able to prevent ALS in the future. So there's a lot of benefits for genetic testing. There are potential drawbacks as well. And so absolutely recommended that you get genetic counseling both before and after. Before, so that you can understand and consider and really think through your decision of getting genetically tested. And after, so that if there is a mutation, the genetic counselor can speak to you about what that means, if there are trials that you can participate in, or if there are therapies that you can get access to. These genetic counselors are trained to help people understand their medical history, the genetics, the potential drawbacks, or the impacts of what genetic testing does. So I just told you about genetic testing and counseling. Well, where can you get that done? You're probably wondering, how do I get access to that? Well, the very first thing you need to do, if you are a person living with ALS, is to talk to your physician and your doctor, consult with them. And your doctor needs to order, a healthcare professional needs to order the genetic test. So start talking to your doctor, ask about whether testing is right for you, ask for a referral to a genetic counselor. A lot of our multidisciplinary clinics may have genetic counselors already. But if there aren't any, there are genetic counselors that you can make, that you can get access to. And the good thing about the program that has been set up by Biogen called ALS Identified is that that program allows you for free genetic testing. By the way, this is both for people living with ALS, but also their families. So if you're a family member of someone who has one of these 40 genes, you're also eligible for free genetic testing and post-test counseling. And this is available through this program, a diagnostic company called Invitae. But all of that has to get started at your doctor. So talk to your doctor first about getting this genetic test and counseling done. So now you're wondering, we talked a lot about genetics. We talked about, go back to that pie, the first slide I showed you, 90% sporadic or idiopathic, 10% is familial or genetic. And I gave you a couple of examples about antisense therapies that are targeting those genes, one of those 40 genes. I talked to you about SOD1 with tofersen, or FUS, which is fusin. But how does antisense therapies help with sporadic ALS? How can it help that 90% of people where we may not know what causes ALS, but we know that in those sporadic cases, almost 98% of people have what we call TDP-43 pathology. This is the pathology that essentially happens because the TDP-43 protein is dysfunctional, and it starts aggregating and accumulating in cells. And so if we can target this protein or components of this protein or interactors of this protein, then we could go after sporadic ALS or all ALS. Whether you have genetic ALS or not, you can go after TDP-43, you go after almost 98% of ALS. And that's where the new research has been focused on, that people are developing these antisense oligonucleotides for things that are associated with TDP-43 aggregates. And those are things like ataxin-2, which is an antisense therapy that Biogen is developing called BIB105 that's targeted to this protein called ataxin-2. And what we've seen is that those antisense oligonucleotides that stop the production of ataxin-2, in other words, reduce ataxin-2, you can reduce TDP-43 accumulation or aggregates in neurons. And similarly, Curalis is developing a QRL201 compound, which is a Stathmin-2 targeted antisense therapy. And that one is a splice modulating antisense oligonucleotide. Remember, I talked to you about two of them, a knockdown approach and a splice modulating approach, where the splice modulating approach is what Curalis is using to essentially correct the RNA mistakes that are caused by TDP-43 to restore normal protein levels. And that study, both of those studies, the Biogen study for BIB105, for Ataxin-2, as well as the Curalis study for Stathmin-2, are both in early stage phase 1 trials. So what are the future directions? The future directions are amazing. Now that we have a drug on market, we know it works. We know it reduces the protein. We know it has clinical benefit, and it is FDA approved. Yes, we're checking whether it can prevent ALS in people that have the mutation but don't have the disease. But what about the other 40 genes? I talked to you about 2 antisense therapies, once against FUS, once against SOD1, but there are 38, 39 other proteins that we can go after. And so hopefully this technology can be leveraged against those proteins. And increased participation in genetic testing will only help, because the more we do testing, the more we do sequencing, we're going to find more genes that are linked and associated with ALS. And as I said, it's important to look at other proteins that are impacted by TDP-43, because we know that 98% of people living with ALS, regardless of which type of ALS they have, sporadic or genetic, they have TDP-43 accumulation. So if we can use this therapy to go after TDP-43, that would be good. And this is just summing up sort of some of the information. I know I covered a lot of ground in the last 45 minutes. I just want to make sure that you had some of these links with you so that you could go back and look at some of this information that I've provided. You could go to als.org/genetics. You could go to als.org/gene-targeted therapies, or als.org/genetic testing. And these will give you links and a lot more information than I was able to cover in this talk. While you're sitting there and thinking, you know, I would like to be part of this, well, we want to hear from you. And we want you to go out and get genetically tested. We want you to go out and be part of clinical trials. But you could also participate in research from home. And you could do that through our ALS Focus study. If you are interested in providing your perspectives and thoughts about how drug development should happen, or what matters to you when it comes to risk-benefit for treatments and clinical trial design, well, you have an opportunity to do that, because we are going to open a new survey through our focus program on clinical trials and treatment needs. And the survey opens April 2nd. And there's a code here that you can scan in, which will take you to alsfocus.org, where you could register so that you can be part of this survey and this really important research to gather your perspectives on drug development and clinical trials in the ALS space. And I finally wanted to end on bringing your attention to a conference that the ALS Association is going to do in the middle of summer, in the middle of Texas, and that is called ALS Nexus. This is the first ever conference in our space that is going to bring care services, advocacy, and research all together under one roof or one umbrella of a conference. And this is a way for you to connect and collaborate with other people living with ALS, with researchers, with clinicians, with people, healthcare professionals. And if you're interested, please visit alsnexus.org. There's an opportunity for you to attend in person. or a virtual option is also available for people living with ALS who are not able to travel or have a conflict because of this being in the middle of July. And so we hope to see you there at our conference in July. And with that, I will stop here and take any questions that you may have. And thank you for allowing me to talk some sense about antisense therapy.
Gosh, Calder, thank you so much for all that information. And yeah, it was a lot to cover, but I appreciate that you did it in a way that most of us non-scientific folks can understand. So we've got quite a few questions, and I'm going to start right at the beginning. April was the first who chimed in, and I think it looks to me like she has more of a comment than a question, so I want to read that. I can only use thumbs on hands, more issues swallowing over last year. I'm afraid feeding tube next. I have vascular brain computer interface Medtronic or Synchron Medtronic has emailed me. So I'm not sure if April's sharing some approaches she's taking. If you have any thoughts on that, feel free.
Yeah, April, thank you for your question. While I can't talk about sort of the different clinical features, what I would suggest is speaking to your ALS specialist. Brain-computer interface is a new technology. It's where this technology is put into surgically implanted into your brain, and it allows for you to be able to just use your brain signals to type or speak. It's really the state-of-the-art type technology that's being developed. And if it gets developed further, it would be groundbreaking for our ALS space. So April, thank you for being one of the first to participate in brain-computer interface type research or treatment. But I would suggest speaking to your physician about the symptoms you're having and how feeding tube would work with BCI and so on.
Thanks, Calder. The next question is, with familial ALS being such a small pool of diagnoses, do you feel we, other ALS organizations, I think we and other orgs, are investing more or less? What does research look like for the 90% of diagnosed folks that we know nothing about?
Well, this is a really important question. I think the question being asked is, are we all just focusing on familial ALS? And we're not. Focusing on familial ALS would be like looking under the night lamp for your keys, right? Because that's where the light is. Not that it's not important. Familial ALS is really important. There's about 10% of people, and we don't want to leave them out. And so the research that's happening in familial ALS around genetic sequencing, around development of new gene therapies, where, antisense oligonucleotide treatment, all of those things are really, really important, and they should continue to happen. And the association also is funding research in that. But you're exactly right. We don't want to lose focus on all ALS, everybody. And our research program is funding research that impacts all ALS, whether it's sporadic or familial. We have, currently speaking, about 30 different therapies that we are funding, whether they're gene therapies, whether they're small molecules, whether they're stem cell treatments. They're not just looking at familial genes, they're going after sporadic targets like TDP43 or Statmin or Ataxin. I talked about some of these in my talk. And we're not just investing in different type of approaches or different type of targets. We're also investing in different types of pathways. Not everybody's ALS is going to be the same. Maybe someone has a mitochondrial ALS, whereas someone else has a ALS because of their immune system, or someone else ALS is because of their autophagy system. Autophagy is handling and processing of proteins. And so our research program takes a comprehensive and a more holistic strategic view of the ALS research ecosystem.
Thank you. Is there any similarity between antisense therapy and COVID mRNA therapy?
No, there isn't. Absolutely no similarity between antisense approach and COVID mRNA therapy. It is really important to know, because I know there is a lot of misgivings about the COVID mRNA therapy. The COVID mRNA therapy does not change your DNA. It is not changing your, going and changing your DNA in such a way that, I know that there's been a lot of kind of misdirection and, perceptions about the COVID therapy. How it is important to know that it targets mRNA. So that, I think that is the similar part of how antisense therapies and the COVID vaccine works, is that the target for both of them are the messenger RNA. In an antisense's case, the mRNA target is our human mRNA, the sort of our gene, our mRNA. In the vaccines approach, the target is the viral mRNA.
I appreciate you addressing that. I bet a number of people were thinking that question. Willie Nation asks, how would you know if you have the TDP43?
Yeah, very good question. So right now, there is no test to know if you have TDP43 accumulation. We are working on that. We have funded different ways to measure TDP43. The ALS Association a few years back funded a tracer development. So essentially think of an MRI where you get put in a big MRI machine and you could scan your brain. We had funded a project to develop a tracer or an imaging agent that would image TDP43 pathology in your brain. That project has been very slow moving forward, and those are the types of projects that would be very helpful to understand where TDP43 pathology is. The other way you could measure TDP43, and we have funded this in partnership with Target ALS, is blood test. Could you measure TDP43 protein in a blood, some type of blood matrix, either in plasma or serum? And those studies are ongoing currently, and we hope that something positive comes out of that. But it will be really important if we can get a biomarker assay. Think of cholesterol right now. You go every year to a doctor, they measure you for cholesterol, LDL, HDL, total cholesterol, and it gives you a sense of, how, are you going to get heart disease in the future? Or should you be reducing your diet of, fatty foods? If we had a biomarker like that, especially if we can measure TDP43, which we know accumulates in, 98% of people living with ALS, one, we could potentially diagnose people earlier. Two, we could hopefully look at the prognosis of the disease. Are people going to be rapidly progressing or slow progressing? And three, maybe we use it as a target engagement marker. Remember, I told you earlier about the two drugs that are being developed against ataxin and statin, where if we can develop those drugs and we had a way to measure TDP43, it would be a good way to measure whether we are actually impacting that toxic protein. And so really good question. Thank you.
And there are more. There's some really nice questions in here. The next question says, what would you say to someone who's lost family members to ALS but doesn't know the type of gene? and are worried about the Biogen testing being shared with their insurers. Do genetic counselors deal with that aspect of testing?
Yeah, they do. So first of all, Biogen testing is not directly shared with insurance companies. In fact, it's not even shared directly with people living with ALS. This is why you have to consult with your physician. Your physician fills out a form and then sends out your DNA to be tested. And then the physician and their office gets the result back. And they are the ones who share the genetic test with you. So this is sort of the process by which it works. Genetic counselors do deal with the insurance aspects. The good news is that if you have ALS and you find out you have a particular mutation, we know that health insurance companies cannot discriminate. That was part of both the GINA Act that was passed, as well as the Affordable Care Act. However, there are some life insurance considerations that you need to have, and this is why speaking to a genetic counselor will be very helpful. We want to just let you know that the ALS Association is doing everything we can to reduce barriers to genetic testing. We know that people are fearful of getting a test because they're worried that life insurance companies may either increase their premiums or deny them coverage. And we are working state by state advocating for discrimination against genetic testing by life insurance companies. I actually recently went to the Maryland State Legislature just a couple of months ago and provided testimony for a bill around reducing discrimination for genetic testing.
Hold up, I'm going to try and get to two more questions. I have a feeling that we could go on for another hour. There are two folks who are wondering, are there any links that have been identified between COVID-19 infection and an increase in ALS diagnosis post-infection?
There have been a couple of studies that have come out around looking at COVID and ALS. And to my knowledge, they have not shown that COVID increased ALS itself. But I think both of those studies showed that COVID exacerbated or potentiated the symptoms of ALS. And so, in other words, people with ALS already have, issues with breathing, right? Because motor neurons are starting to die, and it's hard to breathe. And now, on top of that, if you overlay a respiratory disease or a respiratory infection, like COVID, it's going to complicate respiration and symptoms even more. And so my understanding of those two studies, again, they're very small, so hard to really draw large conclusions, is that while there isn't a line from COVID infections to increase in ALS, people with ALS and their symptomatic burden was definitely affected because of COVID infections.
And maybe more to come on that, because really, in that grand scheme of things, COVID is still pretty new, I would think. And really quickly, last question. In the knockdown therapy that you talked about, are there any kind of side effects or bad effects to that? Are there any problems?
Yeah, this is a really important question. So I told you that, DNA, RNA, and protein, those three things are in our cell, and every protein is made in that way. And the question being asked is, well, if you do a knockdown therapy, you're reducing that protein. Yeah, you're reducing the toxic protein, but you're also reducing the normal protein, right? The protein that's supposed to do its thing, its job. And would that hurt the body? And I think this is a really important question. And this is why companies that develop antisense oligonucleotides that are using the knockdown approach, they have to do these safety studies to make sure that because you're reducing the protein, whether it's toxic protein or normal protein, you're reducing the protein, total protein, that you're not going to have a side effect of reducing the protein. That protein may be critical in doing some other function, which now it can't do because you have reduced that protein. So this is why these safety studies are so important. People may wonder, well, why do people do phase 1? Why can't we just move to phase 2? Well, this is why phase 1 studies and phase 2 studies are important, because it's not just efficacy, especially in these types of approaches. You have to make sure that is affecting the gene, that's going after the mRNA. You want to make sure that that reducing that protein is going to be safe and tolerable.
Wonderful. And I know we've gone over just a little bit, but I felt like that was an important question. Haldip, thank you for your time today, for your expertise, for really giving us this information in a way that most of us can understand it. So thank you for all you're doing to make ALS livable. And we all want to express our gratitude for your efforts.
Thank you, Lori, for having me.
Of course. And thank you to everybody who joined us today. I mentioned last month that we have some more great presentations coming up based on what you have been requesting. A couple of those that are included are emergency preparedness with ALS, exercise and ALS, message and voice banking how-tos. I do want to just send another reminder though. If you're looking for information, webinars are a great place to get just general information, but if you're looking for and needing something specific and you're needing it timely, please reach out to your local care services team. They can get you information much quicker than our monthly webinars can. One more reminder that we've got a very brief survey for you to fill out, if you could, to let us know how you found us and what else you'd be interested in. And with that, I'm going to wish you all a great day and hope to see you next month. Take care.